Hello guys, welcome to the channel, and in today's topic we are going to talk about the structure and the function of immunoglobulins. Now immunoglobulins are defined into multiple ways, and we are going to define immunoglobulin into two different parts. The very first part is related to the structural definition, and second is the functional definition of immunoglobulin. Okay, so structural definition is that immunoglobulin are the set of glycoproteins, and functionally immunoglobulins are those group of gamma globulins or glycoproteins which basically protects our body from various type of pathogens, and those pathogens basically includes bacteria, fungi and yeast, viruses and parasites. Whereas if we talk about the glycoproteins, they belong to the family of gamma globulins, and these gamma globulins are manufactured in two areas. First is basically the lymph node, and in lymph node, as you may be aware, that there are various type of different structures. Okay, and among these structures there is one particular structure called as the germinal center. So this germinal center is the region where the formation of gamma globulins take place, and apart from that there is a cell called as the plasma cells. They have the property of producing the gamma globulins. So these are the two manufacturing sites of the gamma globulin related protein molecule. Okay, and if we talk about the serum concentration, we can see that if we electrophorese the serum sample, the very first and the maximum fragment of peak will be obtained for the case of the now this fragment is the or this particular region belongs to the region of the albumin, whereas the region from here to here belongs to the region of globulin. Globulin region basically defined over here comprises of three zones, that is alpha, beta and gamma. Now a majority of the immunoglobulins or IGs are classified under the gamma globulin section, and apart from that even alpha as well as beta portion of the globulin region are now recently been discovered that even they have some sort of immunological properties. Now if we talk about the general structure of the immunoglobulin, then we can see that the structure of the immunoglobulin is basically a Y type of structure. So it is the basic structure. This Y-shaped of a structure comprises of different region. This region is called as the light chain. This is called as the heavy chain region. And apart from that, these curvatures which are present over here, they are called as the FAB region, and this portion is called as the FC region, and this white color structures are called as the uh, disulfide linkages, which basically binds the entire structure. So disulfide linkage is basically present in two layers. uh, First one, that associates heavy chain and light chain, and also called as the heterodimer, whereas this region represents the heavy chain and heavy chain binding, called as the homodimer formation. Okay, and if we have a closer look to its molecular structure, we can see that this structure is basically associated with the comprises of light chain, heavy chain, heavy chain hypervariable regions, then hinge region, and apart from that, intrachain disulfide bonds, which is present in the form of the yellow color over here. Then this portion also called as the FAB region. This portion called as the FC region. FAB region is the region where the antigen binds, and FC region is the region for the crystallization, which is responsible for the biological activity mediation. Uh, what it means, we will just say it in the later segment. And now here there is other type of classification. So here VL, CL, VH, CH, such type of nomenclatures are there. V stands for variable. Now why it is called variable and why C is called constant, we will see. So VL stands for variable light, CL stands for constant light, VH stands for variable heavy, and CH stands for constant heavy. Now we will try to understand two concepts behind it. Very first concept is that why one chain is variable and why one chain is constant. So the variability is because those regions comprising of the V or the variable region comprises of at least 110 amino acids. Okay, and those 110 amino acids are basically like basically associated with or varies. It basically varies uh, in different antibodies or different IGs. ठीक है. So Variable region basically represents that the presence of this amino acid, 110 amino acid, which basically changes in different type of antibodies. And second thing is that why it is called constant. It is called constant because these amino acid residues is univalently present in all type of immunoglobulin, whether it is like A, whether it is like D, whether it is E, whether it is G, whether it is M. The amino acid and its number, they are basically constant for each and every immunoglobulin, and there is no variation. So that's why those regions are basically called as the constant region. So that's why there we have variable light, constant light, variable heavy, constant heavy, right? Now secondly, we are mentioning over here that light and heavy, right? So light chain is basically the type of assembly of amino acid groups present on the immunoglobulin, and its molecular weight is like around 25 kilodalton, whereas in case of heavy chain it is around 50 kilodalton. So that's why one region is light and the other region is heavy in its nature. Okay. Now one important thing over here is that the variable regions, that is VH and VL, both of these regions are responsible for complementarity determination, and therefore they are also called as the CDRs, or complementarity determining regions. So CDRs. Now if we talk about the constant region, they are classified into two parts, that is the CH1 and CH2. Okay, so why is it classified so? It is basically classified because CH1 is close to the amino terminal, whereas CH2 is close to the carboxy terminal. Except for CH1 region, none of the other CH group will possess the first nomenclature, whereas those groups which are closer to this carboxylic terminal, they will be definitely designated with the uh, proceeding numbers, that is CH2, CH3, CH4. That means that they all are close to the carboxylic group and not and they are not close to the amino group. Okay, so now we are going to talk about the if we talk about the enzymatic reaction, it means that whenever we treat immunoglobulin along with enzyme, then it basically reacts in different manner. And what is the impact of these enzymes on the structural configuration of different type of immunoglobulin? It can be better understood when we perform the experiment of the enzymatic digestion by using the enzyme 1, 2 and followed by the alkylation reaction. Now the first enzymatic reaction is mediated by the enzyme papain. Second enzymatic reaction is mediated by pepsin, and the alkylation reaction is mediated by beta mercaptoethanol. So whenever we react an immunoglobulin with papain, we will get 2 FAB region plus 1 FC region. This happens because, as I shown you in one of the picture, that the hinge portion, right? The hinge portion basically comprises of the disulfide bond. So the papain enzyme basically acts on this pore. Okay, papain enzyme basically acts on this region, and therefore what it does, it basically starts to cleave it. And whenever the hinge region is broken, so what happens is this FAB region, is separated out as a single unit. So does this FAB region. This is also splitted into one single unit. Okay, so how many number of FAB region we have now? We have two FAB region, and only one constant FC portion will be there. So one FC region will be present. Okay, so in this case, the target is the hinge region disulfide component. And also it should be remembered that whenever we are talking about the hinge region disulfide component, it basically means that it is targeting the those groups which are close to the amino radical, or those particular target molecules which are close to the amino radical. Okay, now if we talk about pepsin, what happens here? If we react any immunoglobulin with pepsin, what happens is it basically targets the disulfide bond, but disulfide bond of the carboxylic region. So what we will get? We will get linked FAB regions, but without 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 FC region. Why does it happen so? Because in this portion the target is the FC region or carboxylic terminal disulfide bond. Okay, so this type of reaction will yield only the FAB region, right? Only FAB regions will be obtained and no FC region will be obtained. Okay, And now if we talk about the alkylation reaction, then in presence of beta mercaptoethanol, what happens is if we treat an antibody with beta mercaptoethanol, then at the end of the reaction, what we are going to get is separate heavy chains, so we will be getting separate heavy chains plus separate light chains. And second reaction is that all of these will contain a sulfhydryl group. Okay, so this is the entire reaction which occurs when enzyme digestion related activity is performed on any of the immunoglobulin. Okay, and apart from that, let's talk about the various type of functions. So the very first function of any immunoglobulin is opsonization. Now what is opsonization? Opsonization is simply defined as the labeling of antigen by antibodies. And what is the impact of this opsonization? So opsonization it induces phagocytosis. It induces phagocytosis. So what actually happens over here is that whenever a foreign body or an antigen like a bacteria, whenever this microorganism enters the body, they usually possesses some sort of unique patterns present on their either body or on their cell wall or on their capsule, fimbriae, anything can act as the antigen. So obviously there are certain molecules present in the immune system even before the entry of the pathogen in the body, which are called as the PRRs or the pathogen recognizing receptors, and PAMPs, called as the pathogen associated molecular proteins. And those particular pathogen associated molecular proteins, they also comprises of certain components called as the opsonin. And that opsonin is nothing but it is the type of the antibody which starts to bind with the antigenic components present on the pathogen, thus imparting a signal to the immune system that pathogen has entered the body and it has to be eliminated or it can cause any sort of adverse reaction, right? So now the pathogen has been labeled and a stimulus is basically sent to the nearby phagocytes. The nearby phagocytes. Now, phagocytic activity is basically possessed by either macrophage or neutrophil. They sense that a pathogen has entered the body and it has been opsonized. It has been opsonized. So the process of labeling of an antigen with the antibodies is called as the opsonization, and through this opsonization certain receptors present on the surface of the macrophages or the phagocytes identify the opsonin related component. So whenever they interact with this labeled antigen which has been tagged by the opsonin protein, interacts with the phagocyte, and then phagocytic mechanism is induced and elimination of pathogen occurs. Okay. Now, second important function associated with is complement activation. So when any antigen binds with the antibody, it forms an complex. Now this complex is recognized by the immune system and they start to release complement proteins. And those complement proteins, when they get activate, then they also eliminate the pathogen by forming pore forming complex, or also called as the PFC, or membrane attacking complex, also called as the MAC. So MAC complex. Okay. Now the third activity or the third important function is called as the transcytosis. Now transcytosis, it means migration of immunoglobulin or Igs. Okay. Example over here is IgA and IgG. So as we all know that IgA is commonly found in secretions. That is the most common place where IgA is found. But apart from that, it can also move to breast milk. So this phenomena is called as the transcytosis. And similarly, IgG, when it crosses the placenta, and that movement is also called as the transcytosis. Okay. So this was all about today's topic, which is related to the structure and the function of the immunoglobulin. I hope so that you might have liked this topic. Don't forget to hit the like button, mention any type of comment if you have any sort of doubt or any suggestions regarding this channel, and don't forget to subscribe it. Thank you.